Okay, good morning, everybody. Happy Friday. So y'all have the third midterm behind you. The sun is shining, and it's Friday. So pretty good. And you guys did really well on the third midterm. I don't think I changed the difficulty in how I wrote questions as compared to previous years. So whatever you're doing, it works. So that's very good. But that said, we do want you to have the opportunity to see get information on the questions that you missed. And so there was an announcement I sent out yesterday, but the review session will be 1 week from today on Friday, right after class. And all you have to do is come up to the 4th floor atrium, and we will be there. And at that time, you can get a list of the questions that you missed, and we can go through them, okay? And so that'll be 11 o'clock on the 4th floor atrium, right after this class. 1 week and 50 minutes from now. So anyway, that's good stuff. But today we're going to get into G protein coupled receptors. And so the way this is going to flow is the following. GPCRs, G protein coupled receptors, are pretty complex, and they do a lot of things. And there are a lot of drugs that target GPCRs. So we're going to spend all of today just going through the molecular biology of G protein coupled receptors. And then on Monday, armed with the knowledge that you will get today, we'll be set up for going through a lot of the pharmacology and in-depth MOAs of drugs that target GPCRs. And we'll look at how those work from the molecular level to the physiological organ-based level. And yeah, it's pretty interesting stuff. So G protein coupled receptors are pretty cool. They have some interesting biology. I'm going to go ahead and dim the lights so that you can see the screen a little better, and we'll get into this. There we go. Okay. So these things are called G protein coupled receptors for a reason. And just to reorient you, we're looking at these guys right here. And so these are receptors with typically 7 transmembrane domains. But when a ligand binds a receptor, that receptor is associated with other proteins called G proteins on the cytoplasmic face of the receptor. And typically, once those G proteins are activated, they go on and then they activate, or in some cases repress, second messenger systems, okay? And so that's what we're going to be getting into. So signal transduction, of course, you've seen this term before, but it's basically everything downstream from the receptor to some kind of a cellular and even a physiological response. So signal transduction by G protein coupled receptors, and I'll probably just say GPCRs from now on. They're fundamental for almost every physiological process you can think of. Vision, smell, taste, a lot of neurobiology, effects on the heart, smooth muscle, endocrine systems, reproductive functions. And the Nobel Prize in 2012 was awarded for 2 scientists, Bob Lefkowitz and Brian Kobilka, but they worked on GPCRs. And the interesting thing is that the human genome has now been completely sequenced for a couple of decades. And if you look at the human genome, you can actually go through and identify the genes for GPCRs. And there are somewhere in the region of 800 GPCR genes. So there are 800 different GPCRs in the human repertoire of proteins that can be expressed. And about 30 or 40% roughly of pharmaceutical drugs target GPCRs. So they're a major class of targets for drug development. And the ligands that bind GPCRs are also diverse. Hormones, especially neurotransmitters, all sorts of things can bind GPCRs. So it's probably the most extensive receptor system, not probably, it is the most extensive receptor system that exists in humans and mammals. Okay, so let's get into what these GPCRs actually look like. So we're first going to go ahead and talk about the actual physical structure, the protein structure of GPCRs. So all of them have 7 transmembrane domains. And here we're showing those 7 transmembrane domains of a GPCR stretched out kind of just for visual display across the cytoplasm. And what you can see is that the N-terminus is always on the extracellular face, ECF. So the N-terminus is always outside of the cell. This is transmembrane domain 1, and then you have a cytoplasmic loop, transmembrane domain 2, an extracellular loop, and 3, another loop, 4, another loop, 5, 6, 7. And so every single one of these 800 or so GPCRs has this general structure. And a couple of things to note. The N-terminus is always on the outside of the cell, and the C-terminus is always on the inside of the cell. But the important thing is this is just kind of stretched out. I mean, no GPCR really looks like that. So that's just so you can see the linear structure of how the transmembrane domains penetrate and loop back and forth in the cell membrane. But when you actually look at it from the top, these transmembrane domains all kind of fold up and interact with each other. So they're really kind of in a circle, kind of a barrel-shaped structure, okay? And that's what it looks like in real life. And so I guess this is looking down from the top of a cell, the outside of the cell. There's that N-terminal tail, and then these different transmembrane domains, which all tend to cluster together into kind of a barrel-shaped structure. And you'll see that extracellular loops 1, 2, and 3, as well as the N-terminal tail, are on the side of the extracellular side. And those loops up there kind of form a pocket for ligand binding, okay? So they're very important for where ligands and where drugs bind. And a similar thing is going on if you view it from inside the cell. These C1, C2, and C3 regions and the carboxy terminal tail all kind of group together as well. And those ones are involved in, for the most part, interacting with G proteins, which I'll talk about in a moment. But right now we're really focusing on the receptor. So once again, here's some more detail on the structure of GPCRs. The N-terminal tail tends to be glycosylated. So that's a post-translational modification. These are sugar polymers that are attached to the N-terminal tail. Ligand binding is interesting because it does involve these loops, but that, as you know, these kind of form like a circular structure. The interior side of these transmembrane domains also provide some ligand binding activity. So once they kind of loop together into this circular structure, there's kind of a cleft in the middle that almost it kind of extends down underneath the cell membrane a little bit, and ligands can bind there. The C-terminal tail, there's another post-translational modification that happens there, and that's palmitylation. So palmitylation is basically a fatty acid type modification, and so it's a hydrophobic modification, and these residues can then penetrate and then kind of get anchored into the cytoplasmic side of the cell membrane. There are also phosphorylation sites present on that C-terminal tail, which regulate the receptor as well, and things like the half-life of the receptor. But then I want to draw your attention now to internal loop 2 and 3. These ones actually interact with another protein called a GTP exchange factor, or GEF. And we'll talk about what that means too. But basically, these are the GEFs is another protein that can interact with GPCRs, and it will regulate the duration of the activity of that receptor once a ligand is bound. Okay. So here comes your first view of what the whole thing really looks like. So here is our GPCR, 7 transmembrane domains, and we're showing this receptor in a basal state and an active state. So basal just basically means a resting state. Ligand is not bound, and this receptor is not doing anything, it's just sitting there. But when a ligand binds, it becomes active, okay? So we're going to get into what differentiates a basal state receptor from an active state receptor. And obviously it's ligand binding, but there's a lot of other stuff going on. In addition to the GPCR receptor itself, shown in blue here, the reason they're called G protein coupled receptors is that G proteins are another class of proteins. They're not receptors themselves, but they are 3 different types of proteins that actually bind to the GPCR. And the G proteins themselves, there are 3 different G proteins called alpha, beta, and gamma. And these, they're all G proteins, but each of them is a separate protein. So sometimes you'll see GPCRs called heterotrimeric G protein receptors. This is indeed a heterotrimer. Hetero because each of these is different, but they form a trimer. So the whole G protein coupled receptor complex consists of a GPCR and then 3 other entities, 3 G proteins, that associate with these loops in the cytoplasmic side. And you'll notice that, interestingly enough, the gamma G protein also has a modification that anchors the gamma G protein into the cytoplasmic side of the cell membrane, okay? So why are they called G proteins? They're called G proteins because they can bind either GDP or GTP, guanidine diphosphate or guanidine triphosphate, okay? So that's a nucleotide, right? And that nucleotide is really important because it's the real mechanism for what turns this on and off inside of the cell. And so now we can kind of address how these things work. So here is a GPCR. Ligand is not yet bound. It's in a basal resting state. Alpha, beta, and gamma are associated with the G protein on the cytoplasmic side, but the alpha subunit, the alpha G protein, is associated with GDP, okay? Now something interesting happens when a ligand binds. When ligand binds, this whole receptor kind of twists its conformation a little bit, and that GDP is now replaced with GTP, okay? So ligand binding induces nucleotide exchange, and GDP is kicked out, and GTP associates with the alpha subunit of the G protein. And when that happens, something interesting occurs. The alpha, beta, gamma heterotrimeric complex of G proteins then dissociates from the GPCR, okay? Means it detaches, and the alpha subunit, now complex with GTP, will go down and start a signaling cascade. And then the beta gamma also can exit, and that starts a different signaling cascade. But beta gamma tend to stick together as a heterodimer, whereas alpha is on its own, okay? And this says modulation of effectors. Effectors are other proteins that do something very important. What they do is they activate second messengers, things like cyclic AMP, okay? So there's a lot going on here, right? But this is really important to know how all of this works. So to summarize, ligand binding causes conformational changes in the GPCR itself, and it causes GDP to be exchanged for GTP. GDP leaves, GTP associates, and then the G proteins dissociate from the receptor, and they'll go on and do other things. This whole thing is also very highly regulated. So as long as this alpha subunit is bound to GTP, it's going to go on and keep producing second messengers, basically, by interaction with these effector proteins. And that would happen forever were it not for proteins called RGS proteins. And it's not defined here, but RGS stands for regulator of G signaling. And what these RGS proteins do is they basically stimulate the hydrolysis of GDP into GTP. So they cleave a phosphate off GTP, and that turns it into GDP. So GTP is turned into GDP by RGS action, and then that G protein is no longer active, and it's free to associate with a non-ligand bound G protein again. So there's a cycle of G alpha. It's associated with GDP, signaling, binding of a ligand, releases it. Now it's bound to GTP. It's making things happen in the cell. But after a certain amount of time, it runs into RGS proteins, and that causes hydrolysis of the triphosphate on the GTP, turning it into GDP, and now that alpha subunit is not active, and it's free to associate with an unbound, an unliganded G protein, coupled receptor again. Okay? So kind of an interesting thing. And remember that there are a number of proteins involved in this beyond the receptor itself. Okay? Another thing to note is that there's a whole bunch of stuff downstream from here that we'll also get into. Just a little bit more about ligand binding. In the inactive state, the ligand is not bound. In the active state, it is bound. And here you can see that it kind of tends to bind into a pocket nestled between these transmembrane domains. And that causes a conformational shift in these transmembrane domains. And you can kind of see that a little bit better here. Here's the inactive GPCR. We're just looking at the receptor itself. But when ligand binds, the whole structure of the GPCR kind of twists and shifts a little bit, and that allows this exchange of nucleotides and activation. If you look straight down on these transmembrane domains, you can see that a little bit better, I think, in the lower part of this diagram. So here is the orientation of the 7 transmembrane domains in a GPCR without ligand bound. But when ligand binds, you can see that these helices assume a slightly different conformation. And sure, it's different on the outside of the cell, but on the inside of the cell, it changes those cytoplasmic regions, and those are the regions that interact with the G alpha subunit of G proteins, and that puts it in a position where that nucleotide exchange can occur. Okay? So some interesting kind of structural protein stuff going on there. So interestingly enough, all GPCRs have the same general structure, 7 transmembrane domains. There are a bunch of families of GPCRs, but if you look at the actual amino acid sequences of these 800 or so GPCRs, they don't have a lot of amino acid conservation, but they all have that same general structure. So they've diverged a lot in evolution, but they've maintained that general structure, even though it would be hard to look for actual similarities in amino acid sequence. The binding site of small agonists is often kind of nestled between the helices, and extends almost across down into the membrane region a little bit, but sort of nestled in that pocket. There are also proteins that can bind to the N-terminus. For instance, GABA, glutamate and GABA receptors, there's sort of a interaction with these receptors have a very long N-terminus, and that is kind of specific to those. But all GPCRs have some common activation features, and they all kind of do a conformational change when ligand binds, which impacts their ability to facilitate GDP for GTP exchange of the G protein alpha subunit. Most of them are monomers, but there are 3 different subunits. Oh my gosh, my font got messed up. This should say alpha, beta, gamma. But obviously there's, that doesn't look like alpha, beta, and gamma to me. But anyway, there are 3 distinct subunits, alpha, beta, and gamma. So people have tried to classify G proteins into families, and I don't find this particularly useful, but I'll show you that because you might see this, class A, B, C, D, E, F, or 1, 2, 3, 4, 5, 6. And, these are rhodopsin-like, secretin-like, metabotropic glutamate receptors, pheromone receptors, fungal receptors, etc., frizzled, smoothened. I just show this because you might run into it, but I think that G proteins do so many diverse things that this classification stuff that you'll see doesn't do that much to add to understanding of it. But just so you know, like for instance, the rhodopsin-like family, basically each of these families, you can find some sequence similarity within a family, but the rhodopsin-like family might have much different amino acid sequence than like these other ones. But you can see that they do all kinds of things, like odorant receptors, GPCRs that allow you to smell different things, monoamine receptors, there's just all sorts of attempts to classify these based on what they do. But I think my personal take on it is that classification stuff has very limited value. Okay, so let's get into what regulates GPCR activity. So you can kind of already guess that there's a bit of a cycle that goes on here, right? And a lot of it's maintained by, there are 2 really important things that happen before I get into this. Nucleotide exchange is one of the important features, where GDP is replaced by GTP, and that turns things on. Well, and what turns it off is when GTP is hydrolyzed back to GDP, okay? So activation is mediated by nucleotide exchange, and deactivation is mediated by nucleotide hydrolysis, dephosphorylation, okay? And so here we can go through this, and you can see this again, how this works. So there's a little key down here. It's a little tricky. Notice that this little guy here is GDP, and here's GTP. If you look closely, there's 3 phosphates on that one, 2 on that. But here's the resting state. Alpha, beta, gamma G proteins are associated with the receptor. Alpha has a GDP associated with it. A ligand or a drug comes and binds this resting receptor, this basal receptor, and you get nucleotide exchange. GDP is kicked out and replaced by GTP. That causes dissociation of the G proteins. And the alpha subunits go along and interact with another protein, and for now we're just calling it an effector. And the beta gamma goes ahead and associates with a different effector, and you get various cell signaling. Incidentally, beta gamma is tethered to the cytoplasmic face of the cell membrane, so it can diffuse along in the membrane from one place to another until it encounters an effector. But all of that release, the diffusion means, in the case of beta gamma, it's still tethered to the inside of the cell membrane, and it can just kind of move along the intracellular side of the cell membrane, but still tethered there by this modification. Okay, but so this is a G-alpha bound to GTP. It's signaling via an effector that's going to have something to do with second messengers. But RGS, regulator of G signaling proteins, then can associate with this, and it causes hydrolysis of GTP. That means it becomes GDP, and then it gets turned off. And then the whole complex can get together again for a resting state. So that is the activity cycle of GPCRs. And yeah, any questions on that? Some kind of complicated molecular biology, but I'm trying to explain it the best I can here. Okay, so once again, just a little review. Ligands bind to the transmembrane domains. There are just so many different ligands, biogenic amines, dopamine, epinephrine, chemokines, even some lipids can bind, other small molecules. Okay. And we'll get into GEFs a little bit later. That's, GTP exchange factors. So there are RGS proteins, and there's another regulatory protein called a GEF, but that's shown better somewhere else. All right, now comes something quite important. So far, we've just shown you G proteins here, alpha, beta, gamma G proteins. It turns out there are about 20 different versions of G-alpha G proteins, okay? And so each individual receptor is associated with one and only one type of G-alpha protein. But there are 20 different types of G-alpha proteins. And one receptor might interact with an alpha protein called GI, and a completely different GPCR might associate with the GS. And so there are about 20 different G-alpha proteins available, but each receptor is kind of idiosyncratic. It associates with a particular type of G-alpha. But those 20 G-alpha proteins can be divided into 4 different families. And here I think the family designation is quite important. So these 4 families of G-alpha proteins are called G-alpha I, G-alpha S, G-alpha Q, and G-alpha 12-13. So GIs, that G protein, once it's released from the GPCR, it encounters an effector enzyme. Actually, it's adenylate cyclase. That's the enzyme that makes cyclic AMP, probably the most famous second messenger. GI inhibits cyclic AMP. So it's going to prevent the production of cyclic AMP. But even so, that can result in the opening of channels, for the most part, potassium and calcium channels. If it's a GS type of G protein, it stimulates adenylate cyclase, and it makes cyclic AMP, okay? So this one's inhibitory, and this one's stimulatory. GQ type G-alpha proteins activate phospholipase C, a different second messenger system. And G12-13s activate another signaling pathway which is mediated by Rho GTPases. Okay, so it's a wholly different pathway. Probably the top 2 are the most common. GQ is pretty common, this is less common. But it's interesting that GI and GS do really opposite things, don't they? One inhibits adenylate cyclase, the other activates it. So remember, it's a heterotrimer, alpha, beta, gamma. The beta gamma subunits also are very potent signaling molecules, and they can go on and do other things, engage different second messengers, but they have a tendency to do things such as regulating voltage-gated calcium channels, affecting neurotransmitter functions. So one GPCR can do a couple of different things because of this. So this is another good diagram here showing you basically that alpha and beta gamma do two different things. So ligand comes and binds a GPCR, and then the alpha subunit can go and start a signaling pathway, which is mediated by forming or inhibiting second messenger systems. At the same time, the beta gamma heterodimer can go along and do other things, like opening and closing channels, for instance, or regulating PI3 kinase, okay? So there are also some regulation, the fact that this is phosphorylated on the C-terminal tail, there are other systems that regulate the half-life of the GPCR itself, things like arrestin and GRK. This is a kinase, I think, that can phosphorylate that, and arrestin is different. But the main point of this diagram is that alpha does one thing and beta gamma can simultaneously kick off other signaling cascades. And for instance, when a G alpha S associates with its effector, if that effector is the enzyme adenylate cyclase, that will turn ATP to cyclic AMP, okay? So the effector is adenylate cyclase, and G alpha S will stimulate that, whereas G alpha I will inhibit that effector, okay? All right, so now you know about the receptors themselves, the alpha, beta, gamma subunits, and kind of what turns them on and off. And you also know that a ligand binding typically initiates two independent signaling cascades mediated by alpha and then beta gamma. But we need to really understand more about the second messenger systems themselves to really see what's going on here. And so you can see we're slowly kind of working downstream down this signaling pathway to see what happens. Okay. So now we're going to see the whole signaling pathway for the first time here. And in this case, as an example, we're looking at dopamine receptors. And there are a bunch of different dopamine receptors. But it doesn't matter what they are. What I want you to notice, though, is that here's a receptor called D1, here's one called D5, here's a different receptor called D2. It turns out that these receptors, either D1 or D5, those are GPCRs, associated with alpha, beta, gamma, but D1 or D5 are associated with an alpha, beta, gamma that contains an alpha S G protein, okay? So when ligand binds here, that alpha S will dissociate, it will activate adenylate cyclase, and it will turn ATP into cyclic AMP, okay? And cyclic AMP is a second messenger. Once cyclic AMP is formed, it goes along and it activates kinases, protein kinases. And that in itself might do some quick signaling in the cytoplasm. But ultimately, that kinase might also activate a transcription factor and cause it to translocate into the nucleus. And then that transcription factor can bind to a regulatory element on a gene and do things, but ultimately the transcription factor will cause transcription. You make a messenger RNA, and then basically you make a protein. And you can see this protein comes along and regulates the gene further. But you have this early response mediated by kinases in the cytoplasm, and a kind of a later response involving changes in gene expression. But that whole signaling cascade is kicked off because D1, for instance, is associated with a G-alpha S that stimulates the effector enzyme, in this case adenylate cyclase, to produce cyclic AMP. And that second messenger goes on and does a lot of other things. In contrast, here's this receptor D2, but unlike D1, it's associated with an alpha I. So ligand binding here is going to decrease the activity of adenylate cyclase. It's going to inhibit the production of cyclic AMP, and all this stuff will get turned off, right? And so, adenylate cyclase is what's really doing the work here by making the second messenger, cyclic AMP, and some receptors are stimulating it, and other receptors are inhibiting it. So if you think about that in terms of biology, the balance of ligands that bind D1 versus D2 are going to be regulating whatever that cellular function is, okay? And so there's a lot of dynamic stuff going on. That's pretty interesting stuff, okay? And this D2 with an alpha I has the opposite effect of D1 that has an alpha S. So knowing the identity of the G protein types that are associated with the receptors is completely critical to understanding to how they're going to function. There's some other regulation that I should mention briefly. So here's ATP, adenylate cyclase acts on ATP, and now you have cyclic AMP. You can see the cyclic phosphate here involving the 3' and the 5' carbons in the ribose sugar. But cyclic AMP itself, the second messenger, also doesn't exist forever. There is another enzyme, phosphodiesterase, that turns cyclic AMP into 5' AMP. It basically cleaves off the cyclic part. And that is no longer a second messenger. So the half-life of the second messenger itself is also very important. And that's mediated by phosphodiesterase. And incidentally enough, there are a lot of drugs that are actually phosphodiesterase inhibitors. What do you think a phosphodiesterase inhibitor would do? If you inhibit this step here, you get accumulation of this, right? And so phosphodiesterase inhibitors would cause an increase in the amount of cyclic AMP, and you might get much more cell signaling. It would probably also be a very generalized response because cyclic AMP participates in activating so many signaling pathways. Okay, so signaling, what is it? Ultimately, it's what the cell does after experiencing a ligand signal. And there are many important features of this, and the whole field is called signal transduction. And oftentimes, one feature of them is that there are cascades of kinases, a protein, a kinase, activating another kinase, and then eventually activating a transcription factor or opening a channel or something like that. But a lot of these kinase pathways are triggered initially by the second messenger, cyclic AMP. That's kind of what starts it off. It's what lights the fuse. For example, beta-adrenergic agonists activate adenylate cyclase, makes cyclic AMP. That phosphorylates proteins like myosin light chain. So this is affecting muscle activity. Most of, cyclic AMP is a great second messenger, but I also want to mention that in some systems, cyclic GMP is a potent second messenger as well. And once again, you have an effector enzyme, guanylate cyclase, and then once again, you have phosphodiesterase that can destroy the activity of that second messenger. And that's a negative regulation. And it's always important to have negative regulation because otherwise, once you turn things on, you can never turn them off. And that's just not how we work. So this is guanylate cyclase signaling. And this is not GPCRs, this is other types of receptors. But basically, you can see that, here's guanylate cyclase making cyclic GMP, and that regulates all sorts of stuff. But when you get an influx of calcium into cells, that can affect calmodulin, and that makes nitric oxide, which is a gas, right? And NO actually regulates guanylyl cyclase. So here you can see two different second messengers participating in this, nitric oxide and cyclic GMP. Okay. So here is another example of a full signaling cascade. And what you can see here is sort of a classic signaling pathway mediated by epinephrine. And so this is kind of not really showing the whole GPCR and everything. But what happens is, and we're really focusing more on what happens inside the cell. But basically, epinephrine binds a GPCR, and it activates adenylate cyclase. This is a G-alpha S subunit. ATP then becomes cyclic AMP. Of course, phosphodiesterase is destroying it, regulating the levels of cyclic AMP that accumulate. And then cyclic AMP is now then activates a kinase cascade. So C2R2 is a kinase. It becomes active. And it phosphorylates a different kinase, phosphorylase kinase. Now you have active kinase. And you're using ATP each time you do this to add it's a source of phosphates. Active phosphorylate kinase then phosphorylates phosphorylase B. That becomes active. So you got three different kinases, and once this one's activated, finally it phosphorylates glycogen to become phosphorylated glucose, and you basically end up stimulating glucose pathways in the liver, okay, and glycolysis. So what are the features here? You got receptor binding up here, activating G proteins, making a second messenger. Then that kicks off a phosphorylation cascade of regulatory kinases in the cell. And then the last kinase activates a biological pathway involving energetics, okay? And if you think about, epinephrine, kind of like adrenaline, what you're gonna need glucose if you, so if a tiger comes along and jumps out behind the Gantt building or something, you're gonna run, right? You're gonna need glucose to do that. How does your body mobilize all that glucose to, quickly respond to a stimulus like that? Well, this is actually what's going on inside your cells. Okay, so we focused a lot on cyclic AMP as a second messenger. Let's mention a little bit about the phosphoinositide pathway. And so there is another effector called phospholipase C. And basically, it activates inositol diphosphate to become inositol triphosphate, IP3. And it also forms diacylglycerol. So what's going on with that? Well, we can see it right here. So we've talked about G-alpha S and G-alpha I. Now let's talk about G-alpha Q. Okay, so here's a GPCR. This GPCR is associated with the G-alpha Q type. And so when ligand binds, GDP is exchanged, and then you have GTP bound to the G-alpha Q. And that goes along and stimulates phospholipase C beta. So kind of a two-step procedure here. But the important thing is that this then takes this ungodly looking thing here, PI45 bisphosphate, whatever. But the important thing is that this is a resting molecule that actually contains two different second messengers waiting to be activated. And basically, when this second messenger system is embedded in the cytoplasmic side of the membrane. And these are basically effector molecules right here, stimulated by G-alpha Q. I guess this is the G-alpha Q, and this is the effector enzyme. Once that's activated by G-alpha Q, this whole business here, there's a cleavage event, and it makes diacylglycerol, which is a second messenger, and IP3, which is also a second messenger. And this ungodly name up here is what you call them in its resting state. But it's basically inactive second messengers. But once activated via the action of G-alpha Q, you now have diacylglycerol and the second messenger IP3. So this one up here, diacylglycerol, is tethered to the membrane, but it can diffuse through the membrane and activate protein kinase. It's involved in activating protein kinase C, whereas the IP3 can open channels in the ER, endoplasmic reticulum, and release calcium stores. And this also is involved in signaling. So basically, the signaling pathway kind of splits and then comes back together again. But you need both diacylglycerol and calcium mediated by IP3 to then activate this, which is kind of a famous kinase called protein kinase C. And then downstream from this, other signaling occurs. So this is a completely separate signaling pathway mediated by G-alpha Q. Okay. So IP3 and cyclic AMP are two different second messengers, right? And this is activated by G-alpha S, inhibited by G-alpha I, and this is G-alpha Q, generated second messenger. In some cells, those different pathways are complementary. In other ones, they're antagonistic. And so different cell types behave different ways because of how they're set up with their receptor complements. So anyway, there's a lot of complexity. In addition to all the stuff I told you, some cells have certain receptors and other cells have different receptors. Some cells have combinations of receptors that are different from the combinations in other cells. And so cell type A might respond quite differently to a given stimulus than cell type B because of how they're configured. This one I'll just kind of go through really quickly, but lithium used in psychopharmacology actually affects IP3, and this is a complex pathway. I don't think I want you to know the whole thing. But IP3 and diacylglycerol are involved in neuronal things like neuronal growth cone spreading, potentials in the hippocampus, cognitive impairment, stuff like that. So anyway, lithium can definitely, and there's just tons going on here, but it's actually affecting some of these intracellular signaling pathways. Nitric oxide is interesting because it can actually diffuse and affect the cell next door. So nitric oxide synthase is actually the effector enzyme, and it produces nitric oxide. And so once NO is made, since it's a gas, it can diffuse through the membranes and enter an adjacent cell and signal something in an additional cell. And there are some pathways for NO that cause the production of cyclic GMP. So wow, that's interesting. This cell binds a ligand, and then NO is produced, and the cell next door responds. But NO has a very short ability to do this. It'll usually go like 2 or 3 or 4 cells away, but no further. But there's like, this allows kind of crosstalk between cells. Okay. G proteins can affect all kinds of things, channel activities. Ultimately, they can open and close channels. They can affect enzymes involved in signaling pathways. That kind of goes without saying. I do want to mention odorant receptors very quickly. These are GPCRs. They have 7 transmembrane domains. And there are humans have tons of odorant receptors, but basically there's a different receptor for each different chemical entity you can smell, and that's how that works. So the linings of your nasal mucosa just have all these GPCRs. And I just want to ask you to give me 1 minute because I want to mention one thing that's very important. And it's these GEF proteins that I promised to talk about. So GEF proteins stand for guanine nucleotide exchange factors. And so they're called GEFs. And there are other proteins called GAPs, GTPase activating proteins. So you remember that G-alphas, they are regulated by whether they're bound to a GTP or a GDP. Well, within the cell, a GEF protein, that's a GTP exchange factor, it helps to facilitate kicking out GDP and replacing it with GTP. So that's going to activate G proteins. But GAP proteins, let's see, GTPase acting factors actually stimulate the the hydrolysis, I should say, of the GTP into GDP, and so that will turn GTP into GDP and it turns them off. So GEFs are causing nucleotide exchange, activating G proteins. GAPs activate GTPase activity, and you get hydrolysis and turns it off. Okay? So once again, kind of the balance of these two things is going to regulate the activity of the G protein. Okay, so there's a ton going on, and I couldn't really get into the pharmacology because it just takes an hour to describe the whole system. But on Monday, we'll get into how, knowing all this, we'll go into some examples of drugs that target G protein coupled receptors. Thanks for the extra minute, you guys. That last point is important, so, all right.